Hello students, welcome back. Today we are going to conclude human reproduction in part 9. Today's concept is gastrulation. The cells of blastocyst differentiate and undergo various cell movements or morphogenetic movements, as a result of which they get rearranged into three distinct layers called primary germ layers in a double layered structure called gastrula. Gastrula may be formed by invagination, that means a tissue layer folds in or out. So let's see the diagram here. Invagination or evagination. So already in this statement above mentioned that this cell differentiates, undergo various movements or morphogenetic movements. So here invagination, it is a type of a cell movement or a morphogenetic movement during gastrulation. So in that invagination, a tissue layer folds inside towards the inside. The tissue layer is folds. So here the first mentioned one, the straight line. This is a cell layer, and this cell layer folds inside towards inside or maybe outside to form a cell movement or maybe morphogenetic movement. The next morphogenetic movements, that is involution. So it is nothing but rolling in. Cells move over a lip of tissue and into the interior. So let's see in this diagram involution. So in this the cell layer, it rolling inside. That means the cell layer, it is a straight, and during involution the cell layer rolling inside and form a morphogenetic or rearrangement of the cells. And ingression, breaking up of individual cells. So the cells break away from the tissue and migrate as an individual. It is clear. So in this a layer of cells, the some cells are break from that layer and they migrate as a individual cells. Next morphogenetic movement, that is delamination. The sheets of cells sink in. So layer of cells separate from each other more or less as sheets of cells. We know that lamination meaning we are generally used to laminate our certificates and all. So delamination, separation from that laminated. Suppose delamination of the certificate. That means we need to remove the lamination on that certificate. So here also the layer of cells are separate from each other. So here a group of, we are given that a group of cells layers, that is two layers we are given, that cells are separated to each other. So that we will call as the delamination. So rearrangement of cells or morphogenetic movement is due to all these methods, delamination or invagination, involution, and these. And the next morphogenetic movement is epiboly. So a form of a cell spreading in which cells flatten out. This allows them to cover a much larger surface area. So now let's see the diagram. So in this epiboly, the cells, see once see the number of cells in this layer. in this layer of cells, the number of cells in this layer 8, and the same number of cells here, the occupied area is very less. So these layers are become flattened and allows the cells to cover the large surface area. This type of arrangement we will call as the epiboly. And next morphogenetic movement is intercalation. Two cell layers interlace with each other. So that means in intercalation, the two layer of cells are interlace or joined together to form as a single layer. That we will call as the intercalation. And last one, that is convergent extinction. Cells reorganize to form less layers, allowing the cells to extend out from a point. So now in this diagram, there are several layers of cells are present. 1, 2, 3, 4, 5 layers of cells are present. So these 5 layers of cells are reorganized to form less layers. So total these 5 layers of cells or extend and form as a 2 layers here. If you are observed, all these cells are rearranged and they become less layers of cells. This we will call as the convergent extinction. So whatever the terms involution, ingression, invagination or evagiation, epiboly, intercalation, convergent extinction, all these are the rearrangement of cells or maybe morphogenetic movement of cells to differentiate and form a primary germ layers, and that structure we will call as the gastrulation. These are all takes place during gastrulation. Gastrulation marks the beginning of differentiation of tissue. So we can identify this gastrulation process by observing that differentiation of the tissue. So beginning of differentiation, it takes place during gastrulation. So how that tissue are differentiates? So the differentiates into 3 germ layers. They are ectoderm, mesoderm, and endoderm. And these layers give rise to specific organs, and organs are organized to form organ system. During gastrulation, a cavity called archentron or primitive gut also gets formed in the inner cell mass. Formation of endoderm. Already we discussed that gastrulation marks the beginning of differentiation of tissue, and the tissues are endoderm, mesoderm, and ectoderm. So in that, how the formation of ectoderm it takes place during gastrulation. So now, as the blastodermic vesicle, also called as the blastocyst, enlarges, some of the cells from the inner cell mass get detached and pushed into the blastocele. These are the first endodermal cells. Number of such cells appear on the inner side of the trophoblast. These cells divide rapidly and form a complete layer inside the trophoblast layer. The embryo takes the shape of a tube, the inner tube being lined by endodermal cells. This inner tube is now called as primitive gut or maybe archentron, which later differentiates into 2 regions. And now let's see this diagrammatic representation of the formation of endoderm. So we are already clearly said that the blastocyst is also called as the vesicles or blastodermic vesicle. So during this endoderm formation, it enlarges, means the size is increased. Some of the cells from the inner cell mass. Let's see in this diagram part A. In this diagram, this is entirely we will called as the structure is blastodermic vesicle, and these are the inner cell mass, and this layer is tropoblastic cells or tropoectoderm, and the cavity inside the space inside this region, this part is called as the blastocele. So now what we are saying in the first step, some cells from the inner cell mass detached or separate and pushed towards the blastocele, towards this the cavity. So these detached cells are called as first endodermal cells. So that we are seeing in the enlarged diagram here. So this is the just we are taking only up to this part. So this is a tropoblast inner cell mass, and some of the cells are separating, detached from the, and they are moved towards the cavity, that is blastocele. They are called as the first endodermal cells. Now, these detached cells are rapidly divides to form a complete layer. So first they form a small layers and all are joined to form a complete layer. So that layer is formed towards the inside. This is the tropoblast layer, and towards this inside of this, outside is this region, and towards inside means inside the cavity. So towards inside of this tropoblast, a complete layer of endoderm. So see in the third diagram, a complete layer of endoderm will form. Now the embryo takes the shape of tube. That means inside, when this endoderm layer is formed completely, it appears as a tube-like structure inside, and this tube-like structure is called primitive gut or maybe orchentron. Now the remaining mass of cells left in the inner cell mass after the emigration of the endodermal cells is termed as embryonic disc. One side of the embryonic disc is the cephalic region, and the other side is the caudal margin. So let's see the diagram here. So after formation of this primitive gut, primitive gut or maybe orchentron, the remaining inner mass cells, these cells are called as the embryonic disc. So the cells left in the inner cell mass after formation of this orchentron or endoderm layer, the remaining cell mass is called as the embryonic disc. So in embryonic disc, the one margin, this margin is called as cephalic, that means head region, and opposite region, it is called as caudal margin, that is tail margin, and head margin. So after formation of complete primitive gut, the remaining cells of this inner cell mass is called as a embryonic disc. So what is a embryonic disc? So after completion of emigration of endodermal cells from the inner cell mass, the remaining cells are termed as a embryonic disc. So this embryonic disc has two margins, that is cephalic margin, that means head margin, and a caudal margin, tail margin. So the formation of next layer, that is mesodermal layer. So this mesodermal layer starts after completion of formation of endodermal layer. So after formation of the endoderm layer, then the mesodermal layer starts to form. The cells at the caudal margin of the embryonic disc divides rapidly, increase in thickness, and then gets detached. These cells are mesodermal cells that form a distinct layer just above the endodermal cells. So let's see in this diagram. So now mesoderm, it is formed after completion of the formation of this endoderm layer. Then already we discussed in previous slide, the remaining inner mass cells we are called as the embryonic disc. And for this embryonic disc, one margin we will call as the cephalic head margin, and another side we will call as the caudal margin. So now the mesodermal layer starts to originate from this caudal margin of the embryonic disc. So here the cells of this caudal margin of embryonic disc divides rapidly and increases its cells. And these increased cells starts to detach. So this region starts to detach, and they are arranged to form as a distinct layer over the endodermal layer. So formation of next embryonic layer or primary germ layer, that is ectoderm. The remaining cells of the embryonic disc after the formation of mesoderm and the endoderm spread out and form an outer layer called ectoderm. So already we discussed the first embryonic layer formed is endoderm. Over that endoderm, mesoderm is formed from the caudal margin of embryonic disc. After mesoderm formation, whatever the cells are left in this embryonic disc region, they are all form an outer layer over the mesoderm and endoderm. So remember, all these layers, that is ectoderm, endoderm, and mesoderm, are differentiates during gastrulation. And already we discussed some membranes. Embryonic membranes are different, or fetal membranes, that is chorion and amnion, allantois, yolk sac. These are different. They are membranes formed from the fetus before gastrulation. And differentiation of these cells in the embryo blastocyst to form three distinct layers. They are primary germ layers or embryonic layers. Layers are different, membranes are different. Fetal membranes or embryonic membranes. Generally, two names will mention in your NCERT textbook, that is chorion and amnion. And in our explanation, we are mentioned total four membranes, that is chorion, amnion, yolk sac, and allantois. Four we mentioned. That means some two words we are given beyond that NCRT. So embryonic membranes are different and layers are different. Layers are formed during gastrulation, and these embryonic layers further undergo organogenesis, formation of organs. And after formation of organs, they are undergo organization to form organ system, and organ systems are organized to form organism or form an individual. So let's see the different types of organs which are originate or differentiates from these embryonic layers or primary germ layers. So the organs formed from the ectoderm or organs differentiates from the ectoderm: skin, hair, brain, spinal cord, nerves, mouth lining, lens of eye, inner ear, adrenal medulla. These are the organs are developed from this ectoderm, and the organs formed from mesoderm: notochord or skeleton, muscles, heart, blood vessels, kidneys, gonads, that is testes and ovaries, are developed from the mesoderm layer of the embryo. And organs formed from endodermis. So lining of alimentary canal, that is digestive system, respiratory system, liver, pancreas, urinary bladder, middle part of the ear. These are all developed from the endodermis of embryonic layer. So all these organs are formed and organized to form organ system. So that is brain, spinal cord and nerves all are organized to form nervous system. So respiratory system, that is nasal cavities, nostrils, pharynx, and trachea, larynx, bronchi, bronchioles, lungs, all together form as a respiratory system. That is our differentiates from the endodermis. So digestive system part, that is liver we will discuss under the digestion. Pancreas we will discuss under the digestion. Alimentary canal, it is a part of digestion. So now all these are formed as a digestive system. So first organs are developed from these layers, embryonic layers, and all these organs are grouped and to form as a organ systems, and all these organ systems are organized to develop a new individual. So this is a simple representation of this all the primary germ layers which are formed during gastrulation. So now zygote undergo cleavages and to form it undergo blastulation, and finally it comes under gastrulation. During this gastrulation, the, it is a differentiates into the three germ layers and their functions. So now gastrulation form the ectoderm, mesoderm, and finally endoderm and their organs. So outer epithelium of body and its derivative. So ectoderm forms the outer epithelium and its derivatives: hair, nails, epithelial glands, lining of mouth, enamel of teeth, lens of eye. So in previous slide we mentioned some organs, And here in this sketch, we can get some more organ information. So lens of eye, inner ear, nasal and olfactory epithelium, skin, epidermis. Next, neural tube, that is brain, spinal cord and motor nerves, and neural crest, sensory ganglia and nerves, adrenal medulla, sympathetic ganglia, skull, gill arches, dentine of teeth, all from the neural crest. So simply from ectoderm, outer epithelium of body and its derivatives, neural tube and neural crest or differentiates. And then from mesoderm, notochord, lining of thoracic and abdominal cavities, circulatory system, uh, somates and organs of urogenital system are differentiates from the mesoderm. So now circulatory system, blood, bone marrow, lymphoid tissue, endothelium of blood vessels, lymphatics are developed from the mesoderm. So somatis. So that is skeletal muscles, bone, cartilage of skeleton, except skull. Skull is formed by ectoderm. And dermis and connective tissues developed from the mesoderm. Organs of urogenital ducts, that is ureter, kidney, gonads, reproductive ducts developed from the mesoderm. And endoderm, from this a primitive gut is developed. From this primitive gut, epithelium of respiratory tract and pharynx, liver and pancreas, epithelium of urogenital system are developed from the primitive gut. So now here the pharynx, again it is pharyngeal pouches, thyroid and parathyroid glands developed from the endoderm layer. Liver and pancreas developed from endoderm layer. Epithelium of urogenital tissue, epithelium, urogenital tissue is covered with a epithelial tissue. Generally, epithelial tissues are helpful to cover over the organs. So epithelium of urogenital system is formed or developed from endoderm. So this all information is helpful for your competitive examinations. From which organs differentiates from which type of germ layer. For that, this representation is essential to quick reference. The next concept, that is neurulation, that means a development of a neural tube. After gastrulation, the process of tissue differentiation begins. During neurulation, the development of neural tube takes place. The neural tube later gives rise to nervous system. Anteriorly, the neural tube is differentiates into brain, and posteriorly, the neural tube differentiates into spinal cord. Gradually, the process of organogenesis begins. First, the organ rudiment appear, which eventually develop into functional organs. So rudiment means undeveloped or not well developed, and later in development and they become functional. The development of human embryo. The human embryo takes about 9 months or an average of about 40 weeks from fertilization till birth. This time is commonly divided into three periods. They are first trimester, second trimester, and third trimester. The first trimester. So let's see what are the changes we can observe during this period. First trimester, it is from first month to third month. It is first 3 months, which is considered as first trimester. So now it is a period of rapid cell division, cell differentiation, and organogenesis. So after first month, the embryo heart is formed. So heart formation is occurred after first month, and at the end of second month, fetus develops limbs, that means hands and legs, and digits, that means fingers, develop at the end of second month. And at the end of third month, most of the major organ systems are formed. Most of the major organ systems, limbs, external genital organs. Suppose if it is male, penis is the external genital organ. If it is female, we will study that vulva, that is mons phoebus, uh, vagina, clitoris. So these are all formed at the end of third month. And basic structure of baby is formed completely in the first trimester. During this period, first trimester, the developing baby called fetus. So that's why after completion of 3 months of pregnancy, the embryo is called as the fetus because the basic structure is formed. Hence, it is considered as the fetus. By the end of first trimester, all body systems of the baby are formed. All body systems, digestive, respiratory, nervous system, circulatory, reproductive, all system, endocrine gland system, all systems of the body formed at the end of the first trimester. Means at the third month of pregnancy, the embryo got a basic form with all body systems. Hence, from third month of pregnancy, the embryo is considered as fetus. Here we need to know some important thing for this pregnant women. Certain medicines can have highly undesirable effects the pregnant woman. In 1960s, many pregnant women took a medicine called as a thalidomide to minimize the discomfort during pregnancy. The most discomfort during pregnancy is vomitings and not taking more amount of food because the ovaries and the uterus is situated in the abdominal cavity. So when the embryo sizes increases, some discomfort between the organs will develop. So that may create some vomiting sensation and all. So to minimize that discomfort during pregnancy, the most of the pregnant women in 1960s, they are start to using a medicine that is a thalidomide. So it resulted, the thalidomide resulted in many malformed babies. Malformation, that means lack of one or more body parts. Malformed babies, the chemical agent that lead to development of malformed babies are called as the teratogens. So what is meant by teratogens? The chemical agent that lead to development of malformed babies are called as the teratogens. So not only this thalidomide, and some of the women, they are habituated to this tobacco, nicotine, or maybe alcohol. This is also may lead to this teratogen malformed babies. Second trimester. So this is fourth, fifth, and sixth month of pregnancy. That period is called as second trimester. Second trimester is a period of tremendous growth and development. Already we said at the end of this third month, the embryo or fetus gets the basic form with all the body organ systems. So now that organ systems are undergo a tremendous growth and development during second trimester, that is fourth, fifth, and sixth months. So during fifth month, the first movement of fetus can be felt by mother, and appearance of hair on the head we can observe during fifth month. And at the end of sixth month, fetus body is covered with fine hair. Body hair is developed during the end of the sixth month, and eyelids are separated and eyelashes are formed at the end of sixth month of pregnancy. Third trimester, that is seventh, eighth, and ninth month. These 3 months together. It is the third trimester. Third trimester is the period of growth rather than new development. So there is no any new developments during this trimester, seventh, eighth, and ninth month. You all may know that some of the babies are born at the month of seventh or maybe eighth, even though most of the babies which are born in seventh and eighth month, they are also survive because all the organ systems are developed at that time. The only thing is that the further new development will not take place. So for such babies, we will kept few days under incubation. So at the end of ninth month, fetus develop and is ready for birth with all its essential organs well formed and able to function. The next concept, birth or parturition. The average duration of human pregnancy is about 9 months, which is called the gestation period. That is from fertilization to till birth. That period we will call as the gestation period. Birth or parturition begins with a series of strong rhythmic contractions of the uterus called labor pains. The birth of fully formed fetus, the birth of fully formed fetus is called as parturition. This is one of the important short answer. Parturition is induced by, that means birth of fully formed fetus is induced by complex neuroendocrine mechanism. That means involvement of nerve system as well as endocrine gland system, that is glands, endocrine glands. So complex neuroendocrine mechanism giving birth to new baby is a complex neuroendocrine mechanism. The signals for parturition, birth, originate from the fully developed fetus and the placenta, which induce mild uterine contractions called fetal ejection reflex. This is the first step to initiate this parturition. Fetal ejection reflex. It is developed by the fully developed fetus and placenta. Is induces that mild uterine contractions that we will call as the fetal ejection reflex. It triggers the release of oxytocin from pituitary gland, posterior lobe of pituitary gland. So oxytocin, it is released by pituitary gland. That gland is belongs to endocrine gland system. So that's why the mechanism of this parturition is neuroendocrine mechanism. Neuro means fetal ejection reflex, that is a part of nerves that triggers the release of this oxytocin from the posterior lobe of pituitary gland. So oxytocin acts on the uterine muscles and causes stronger uterine contractions, which in turn stimulates further secretion of oxytocin. So first initial mild uterine contraction developed by fetal ejection reflex, that is by fully developed fetus and placenta. Once the oxytocin is released from the pituitary gland and it stimulates the further secretion of oxytocin. So that's why this oxytocin hormone is also called as the birth hormone. Now, oxytocin from pituitary gland and prostaglandins from placenta are the two hormones that actually induce contractions of uterus. Lactation. So during this parturition, oxytocin develops the contractions in the walls of uterus. As a result, the fetus is expelled out through birth canal, through vagina, and this cervical canal form as a birth canal. Through that birth canal, the fetus is expelled out. So now we need to discuss about the lactation. So what is meant by a lactation? The production of milk in the mammary gland is called as lactation. The females mammary glands undergo differentiation during pregnancy and start to produce milk towards the end of pregnancy and after the birth of young one. Secretion and storage of milk generally begins after birth of young one, usually within 24 hours under the influence of hormone prolactin secreted by anterior lobe of pituitary gland. So secretion and storage both are influenced by the hormone prolactin, that is secreted by anterior lobe of the pituitary gland. But the ejection of milk from mammary gland, ejection comes out. Ejection of milk is stimulated by the hormone oxytocin released by posterior lobe of pituitary gland. So both prolactin and oxytocin are involved in the release or secretion, storage and ejection of the milk. So secretion and storage is influenced by the prolactin and ejection of milk is influenced or stimulated by oxytocin. Colostrum, the milk which comes from the mammary gland of the mother just after childbirth for 2 or 3 days is called colostrum. So this colostrum is a yellowish fluid and it contains antibodies. That antibody name is IgA, immunoglobulin A type antibodies present in the colostrum or in mother's milk. These antibodies provide a passive immunity to newborn infant So that is the reason why the mother milk is more essential for the newly born infant, because the mother milk consists antibodies which are required for development of passive immunity, that is development of resistance against the disease causing organisms. And moreover, the colostrum is a yellowish fluid due to presence of more protein. So mother milk consists more protein, that is called as the protein is called as casein protein is present, and mother milk there is no content of fat in the mother milk. No fat, more protein content. Protein is required for the muscle development, tissue development. So you may study that in first PU, tissues, the muscle or maybe tissue. It consists a protein called as collagen. So the newly infant, the muscle development or tissue development required protein. That protein content is more in colostrum, mother's milk. And one more thing, there is no fat in the colostrum. And the most essential thing is that presence of antibodies which are essential for passive immunity resistance against the pathogens. Thank you, students. The human reproduction chapter is concluded. So we will discuss objective part from this human reproduction soon.